Welcome back, ladies and gentlemen. So today we're going to wrap up talking about evolution, really just sort of going back through and specifically talking about the evidence for evolution. Keep in mind that evolution is a theory, and by scientific definition, that means there's copious amounts of evidence to support this as a scientific idea. So where do we draw evidence from? Really it comes from a variety of sources, which is what gives evolution such credibility as a scientific theory. So we'll sort of refresh ourselves, talk a little bit about those fossil records and radiometric dating again. We'll talk a little bit more about genetic information, and that'll also come up when we get into some of the classification schemes and the way we start tracing genetic lineages, evolutionary lineages, distribution of organisms, as well as some anatomical and developmental similarities across species as well. So let's take a look at your fossil record. So we talked quite a bit about this when we were talking about Earth history. So just remember that a fossil is the remains or traces of an organism that died long ago. And so the fossil record is the ordered array of which those fossils show up in the layers of sedimentary rock. So remember when we look at these, assuming that these are in the same orientation that they were when they were deposited, everything down here gets layered first and things layer on top of them as they go, right? Water washes that sediment into an ocean or a calm lake, and all that sediment falls to the bottom, and more sediment continues to fall on top. So any of the organisms that happen to get trapped in that layer, or any remnants of those organisms that get trapped in those layers, have the potential to become fossilized based on the pressure of additional sediments layering on top of that. And eventually that turns into what we know as sedimentary rock. So these show all sorts of different types of organisms. They give us an idea approximately when they would have appeared on Earth based on index fossils, based on the layers themselves. And then we can also talk about being able to actually radiometrically date and get a much more accurate time frame as opposed to just looking at those layers and getting approximate times. And remember that radiometric dating takes into account different radioactive isotopes, right, and the half-life. So I know when that rock initially forms, it has 100% of that particular radioactive element, and that it breaks down in half at a very specific rate, or what we know as its half-life. So by the time it gets through one half-life, we know the time frame that it has passed there. If I'm down to a quarter of it, I know that how much time has passed, so on and so forth, and I can calculate that based on the known decomposition times that it takes for half of that element to disappear from a sample, aka its half-life. Now keep in mind this fossil record only does so much for us. First, we have to be able to discover the fossils in the first place. Secondly, the history of life period is just incomplete in general. Because those organisms needed to have been close to water, they needed to have parts that were capable of being fossilized, they needed to have actually fallen into that sediment and get covered up fast enough before they got drug away by scavengers, so on and so forth. So there's a lot of missing pieces, but when we can find those transitional fossils, something like the Tiktaalik you read about, that really is exciting because it gives us some of those stepwise progressions when we can find them. It gives us some of this ability to see those changes over time. So the study of those fossils themselves gives us a really strong piece of evidence for evolution when we can find those transitional pieces. So something like the organisms here, where we've got these land organisms actually going back into the ocean. So this is how we think the evolution of whales may have occurred. So they initially started off as water organisms, got onto land once we sort of terrestrialized life on Earth, and then they actually went back into the oceans, which is kind of interesting. And we'll talk about why whales have a pelvis, because we think it's connected to the fact that they would have had to be on four legs at some point in their evolutionary history, which is kind of cool. So just some examples of fossils, right? These are hominid skulls, and hominid is human related, so not necessarily human per se, but sort of our ancestors, and where's the connection from the lineage that ultimately goes into monkeys and apes versus the branch that then eventually becomes human. Petrified trees. These are fossilized trees, which are really kind of cool because we can take a look at those ring structures. How big are they? Um, and be able to understand something about the environment, how much water might have been around at the time, how dry was it, so on and so forth, which is pretty cool. These are casts, ammonite casts, so rather than it being the actual organism itself, these guys left depressions in the rock, and so we don't actually have the ammonite itself, but we have its leftovers, if you will, its sort of impression. Think of it like putting your foot in sand and your footprint never disappears. That's kind of what's going on here. Um, Same thing over here. So fossilized organic matter in the leaf itself left the depression, and we can now see vein structure and all sorts of things, which is pretty awesome. These are some more unique fossils. Most kids think about fossils in rocks, and overwhelmingly that's where we find them. But we can also consider things that are preserved in amber. Sap, right, from trees captures the entire organism. And these are really exciting because not only do we just get, unlike the sedimentary rock fossils, where those things are sort of turned into rock or they're just impressions of the organism left behind, this is everything. Soft tissue, potentially DNA, all sorts of really cool stuff when we get these organisms to fossilize in amber because it doesn't require the pressure and the intense geologic processes that create most of the fossils kids think about when they come into this for the first time. Um, and this one's interesting. This is Iceman. He was found in literally ice. And as that ice started to melt, we were he was discovered by a hiker. And again, also really kind of cool because rather than having those intense geologic pressures, just converting his bone into rock, instead, all sorts of soft tissue got preserved so we can do tissue samples, we can do DNA samples, we can do all sorts of stuff, which is really kind of cool to kind of understand what his life was like. All right, so let's talk about molecular evidence as a piece of support for for evolution. And what we're talking about here really are coming down to utilizing three different molecules in our cells. And those molecules are your DNA, your RNA, and your amino acids. I'm going to shorthand this as AA. And remember, amino acids are the building blocks for your proteins. All three of these molecules have a particular sequence within their building blocks. So when we talked about DNA and RNA, remember they are made of those nucleotides, right? And those nucleotides have different nitrogenous bases on them, so A, T, C, G, and U. And so we can put them together kind of like the letters of the alphabet to make different sets of instructions. And your amino acids work the same way, right? We looked at those R groups, and there's 20 different R groups. So the way that I put those amino acids together makes a particular protein based on that sequence. Again, it's like using letters in an alphabet to make a bunch of words. So when we look at this in our cells, there's something that you need to understand, and that is the fact that these three molecules have a connection to each other. So that DNA, right, is sitting in your nucleus, okay? And that DNA never really leaves that nucleus, but ultimately that DNA is what gets passed from parent to offspring, and it is what tells your cells what to do. It is your set of instructions. But if that DNA never leaves the nucleus, how do those sets of instructions actually get into the rest of your cell to tell your cell what to do? And that's where this idea of central dogma or flow of information molecularly exists in your cells. And we'll come back to this later this year and talk in more detail. But for now, understand that that DNA in your nucleus never leaves. So what we do is we make copies of just pieces of that DNA into RNA. Okay, and so we copy the sets of instructions from DNA into RNA. That RNA then can then go leave the nucleus and go out into your cytoplasm, which is everything outside your nucleus. It's where your mitochondria are, your Golgi are, your ribosomes, so on and so forth. And those RNA molecules will then go specifically to your ribosomes. And your ribosomes are your protein factories. Okay. And so what does that mean? That means I'm going to take that copy of DNA that I made into RNA, and then I'm going to go to those ribosomes, and those ribosomes are going to read that copy of information to make the amino acid sequence, aka make the protein. Okay, so all three of these molecules are connected. DNA has the initial set of instructions for every protein your body could ever possibly create. The RNA then is just the copy for that particular set of characteristics I want to create that day, and then we turn that into the protein, which is the actual functional trait, right? So when we talked about those terms, genotype, right, which we've been shorthanding something like this, versus phenotype, which might be brown eyes, all right, the connection here is this is what's coded for in your DNA. That's the actual nucleotide sequence, all right, and you would have a certain sequence for the dominant trait, a certain different sequence for the recessive, and then when we put those sets of pieces of information together, they ultimately create your phenotype, which is the physical expression of that, which is the protein itself. So if I had blue eyes, that means my body makes a different set of proteins because I have a different set of DNA. And hopefully that makes sense. So what we can look at then for molecular evidence are those changes in DNA, right? Which will ultimately cause changes in your RNA, which will ultimately cause changes in your proteins. So if I take a look at those changes, aka those mutations, right? These are changes in DNA, which will cause changes in your RNA, which will cause changes potentially in your amino acid sequence. I can then look at how those species evolve over time based on the changes to a particular trait that they share. So that's what we're looking at over here. So here is a bunch of different species. They all contain this hemoglobin protein, right? Remember, polypeptide is another word for protein, right? Okay. And so we're looking at the percentage of amino acids that they share in common. So if I'm comparing, and this one is a human hemoglobin, human to human, that should be 100%, right? Because we're taking the same protein out of the same species. But if I compare that hemoglobin protein, which is also important to monkeys to function, we share 95% of those amino acids in common. If I compare human hemoglobin to a mouse, 87% of those amino acid sequences are identical. Go all the way down to human versus a lamprey's hemoglobin, and now we only share 14%. And so what this tells us is that over time, we know mutations accumulate. We know that there's sort of a background mutation rate that occurs, and so we can use this kind of like a molecular clock and be able to look at how many changes have occurred over time. The more differences that have accumulated, the more time that has passed since we have shared a common ancestor. The more sequence that we share in common, then the more recent that divergence occurred, the more recently we've split from each other from that common ancestor. So if we were to look at this, humans and monkeys, going back in time, have shared a common ancestor much more recently. I would have to go really deep back into evolutionary history to find that common ancestor that humans and lamprey share. Okay, we can also look at distribution of species and where organisms exist. And this comes back to the idea of the connection between continental drift and evolution. So remember that at one point in Earth's history, all of these land masses were together in this big supercontinent called Pangaea. So over time, as plate tectonics and continental drift have pushed these guys apart, we have seen environmental shifts. We've also seen distribution of organisms. And so when we find organisms that are very similar in places here in South Africa, or excuse me, here in South America and here in Africa, we know that there's a evolutionary connection to them because at one point they used to be directly connected and being able to move from one population here to one population here. As these things have pushed apart, now any environmental change that occurred here changed this species, same thing over here. And so this is the idea of biogeography and the study of these distribution of organisms around the world and how their evolutionary connection is also tied to the fact that Pangaea used to exist. So we can take a look at just the interesting distribution of marsupials that are very much endemic or specific to the continent of Australia, right? And so they got stuck there, and so they evolved very much independently of other organisms elsewhere. The finches with Darwin and the Galapagos are another great example of the fact that there are finch species in South America and then very specific species found on those islands as well, and that's sort of that connection between plate tectonics, continental drift, and evolution. So those pieces of geographic evidence suggest that those different organisms with different plants, with different climates, with different predators, with different food sources, so on and so forth, are going to favor certain traits in the organisms that end up there. And so once these land masses got far enough apart that they could no longer continue meshing together with those other populations, as those environments changed on those isolated places, the species had to adapt along with it. And so we can look at those changes over time as a way to discuss evolution as well.